In this video, we will discuss about next generation sequencing, how the sequencing is carried out in this next generation sequencing. So this is the DNA in the topmost figure. In the topmost figure, we can see the DNA fragment that needs to be sequenced. So first, what we will do is we need to add a fragment with that has this overhang A in it. So we will use the primers in such a way that, or we will use those primers that have this overhang A whenever the amplification of this DNA fragment occurs. And after this, what will have what is the next step? Next step is to add the adapter ligands. And why these adapter ligands are important? These adapter ligands are important because this adapter ligand will help to bind to the uh, oligos that are present on the flow cell of this next generation sequencing. So some part of this ligand are complementary to the oligos that are present on the flow cells, and some part of this ligand are complementary to the this barcode. So half portion is considered as a barcode in order to identify from which sample this DNA fragment comes from. So this is the whole DNA fragment looks like. The, the blue region is the DNA fragment that needs to be sequenced. The pink region is the barcode, and this green and purple region is the different types of uh, adapters that are complementary to the oligos available on the flow cells of the next generation sequencing. And when this DNA fragment with ligated with adapters and barcode are added to the flow cells, it will bind to the this adapters will bind to the oligos that are present on the flow cells, and it can form like this shape. After that, the bending bridge formation occurs because this half part of the top part of the oligo is also top part of this fragment DNA fragment is also complementary to the other oligos that are present on the flow cells. After that, the bridge amplification will occurs. So the primers will add in primers will bind to the oligos. Primers will bind to the this top end of the this is the another part of the flow cells to which this top end of the DNA fragment is binded. After that, what happens is that the primer will help in amplification of this other end of or complementary part of this DNA fragment, and this can be seen here. This clustered of this bridge amplification occurs. After that, what happens is that after this cluster of bridge amplification, uh, we will start to measure the after this bridge amplification, the top part, the newly clustered part will form a straight line, and it can be seen here. And after that, the sequencing of this primer, the sequencing primer is annealed to the top portion of this DNA fragment, and this can be seen here. After that, the same thing that we have discussed in the Sanger sequencing, that is fluorescently labeled oligonucleotide is attached. After that, it will but the difference in this kind of nucleotide is that whenever it releases a signal, that deoxyribose nucleotide dideoxyribose nucleotide will convert into deoxyribose nucleotide, so that after this fluorescence is emitted and signals are recorded, then the further sequencing of this DNA uh, can take place, as it can be seen here. the further extension of this sequencing has been taking place. And simultaneously what happens is that the signals are getting recorded, as one of the example is shown here. The advantage of this and next generation sequencing is that multiple DNA can be recorded simultaneously, and whereas in Sanger sequencing only a single DNA fragment or a single whereas in Sanger sequencing only single DNA fragment can be sequenced at a time. So, and then later on, based on this barcode, the samples can be analyzed uh, and can be separated towards the towards the experiment for which this DNA fragment belongs to. And this DNA fragment, and also we can the barcode. Let me repeat my sentence. The barcode can be used to identify which DNA fragment are being sequenced by this next generation sequencing in a particular uh, read. So that's how this next generation sequencing work. If you need to understand this next generation sequencing in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.